Hi there, back again. I'm going to go through cellular respiration with a emphasis on the oxidative phosphorylation part, but we'll just pass through all of these to get there. So here we go. Let's see if we can progress here a bit on this next slide. All right, we've looked at this a little bit. Just the idea here is we're looking at what the body does when it brings in materials and breaks them down, those catabolic reactions, and then ultimately creating energy for the purpose of any anabolic reactions, such as making more cells or repairing things or making certain products. So that's why that happens. So here we start, though, where we focus only on glucose when we're looking at cellular respiration, but in truth that's not actually the case, because the body is also taking in other types of nutrients, and it's also breaking those down for energy and for the building blocks of other types of molecules. But understand that, but for now we're just going to talk about glucose metabolism. So glucose is going to enter the cell. The enzyme hexokinase is going to be involved with that first reaction you see there. It's a coupled reaction because ATP, the energy from ATP, the release of that exergonic process, is going to push this reaction, which would otherwise be endergonic, forward. And hexokinase is going to take that phosphate off of ATP and add it onto glucose. So there we have glucose 6-phosphate. Now, the purpose for doing that is, for one, to keep glucose in the cell. Glucose cannot then go back through any of the transporters, even when the concentration of glucose starts to build up in the cell. So that addition of the phosphate group is going to capture glucose. The other thing it does is it starts this process of reorganizing glucose in a way that we can start extracting energy from it, oxidizing it, taking some of the energy away, creating a little bit of ATP. So from glucose 6-phosphate, you see we go to fructose 6-phosphate, which is an isomerase reaction. We see that the enzyme involved there is something called an isomerase, basically a rearrangement of the molecule. Reaction 3, again, another coupled reaction. We see ATP there. We see another kinase being the enzyme involved. And we create the molecule fructose 1,6-bisphosphate, just meaning that it originally was glucose. We now made it a fructose with this phosphate group on both carbons 1 and 6. That gets split into 2 by this aldolase. And what we get are 2 somewhat even halves, but one of them needs a little bit more conversion, and that's this dihydroxyacetone phosphate, which is going to be converted by an isomerase into another one of these glyceraldehyde 3-phosphates. So that's that. We follow one of the glyceraldehyde 3-phosphates, or G3Ps, as we call them in class. And right away we see that we have an oxidation reduction reaction. So NAD plus is going to oxidize this G3P, it itself being reduced. And then the end product of that, which you see down below here, 1,3-bisphosphoglycerate, is the next substrate for the following reaction, which is a reaction that involves a kinase. But in this case, notice that the arrow has gone this way. In this case, an ATP is being made. So ADP gets phosphorylated, we get an ATP. And now we have this next intermediate product, which is going to get shifted a little bit more, rearranged. You can see that that's happening along the way. We're going to lose a little bit of water. We get this product at the end here, number 10, which then finally we're going to extract another bit of energy from it. Here's our ATP. So these two reactions that we're seeing here forming ATP aren't coupled reactions, but what we're seeing is energy is getting extracted. The phosphate in particular is getting extracted and making an ATP, And then the other product, other than the NADH and the ATP, is this pyruvate. And so now we're moving on to see where pyruvate's going. Pyruvate, 3 carbons, enters through a transporter into the mitochondrial matrix. Right away, 3 things happen to it. 1, loses a carbon dioxide. Other is that NAD+ is going to oxidize it, the rest of this pyruvate, forming NADH. That's the reduced form. And then we have an enzyme here, a coenzyme, that's going to be involved. And there's another enzyme here, but this is what we're looking at, is this coenzyme A actually gets attached onto the remainder part of this. This is the acetyl group right here, or acetyl group. Here's our coenzyme A. So now put these things together, we have acetyl-CoA. This all happens in the mitochondrial matrix, and you've seen this picture before. Again, mitochondrial matrix, we're going on. Here's our acetyl group with 2 carbons. It's going to be added to this 4-carbon oxaloacetate. That CoA enzyme is going to leave. Now we form citrate, 6 carbons. An isomerase is going to convert it into this little form, so just basically rearranging again, isocitrate. Isocitrate then is going to get oxidized by NAD+, and then as a consequence, NAD+ becomes reduced. Carbon dioxide is removed. Now we're down to a 5-carbon compound, which is alpha-ketoglutarate, which you don't need to know. That also is going to get oxidized. Again, NAD+ is going to oxidize it. Here we go. We have an NADH, another carbon dioxide's lost. Then we get down to something called succinyl-CoA, which you don't need to know, again, but it has 4 carbons. And from here to here, this conversion right here is the first time we're going to see another substrate level phosphorylation, where we have a GDP that forms a GTP, and then that GTP will be converted to ATP. This 4-carbon compound is going to get oxidized further, but this time by FAD, making FADH2. We have a few other little conversion things going on here. We'll go up to here, from malate up through back to oxaloacetate. We see the NAD+ again, so here we see another oxidation reduction reaction. Now we have a reduced form, NADH. Notice no carbon dioxide's lost here, though, because we need to have this 4-carbon oxaloacetate here to again join with the next acetyl-CoA that comes in. There you go. So all of that has happened, as you can see here in the matrix. But now we're going into this inner membrane, where within the inner membrane we have the protein complexes. These are actually enzymes that are these large complexes, 1, 2, 3, and 4, that are going to now take those molecules, NADH and FADH2, and they're going to remove the electrons from them and shuttle them along. And so let's see that in this next picture. So this is the inside, there's the matrix again. Here's the NADH, which comes to complex 1, where the electrons get removed by complex 1. The energy that's released from that will pump the hydrogen ions out into this intermembrane space. Meanwhile, the electrons are going to get passed to the next part of this chain, and this is Q, sometimes called CoQ, sometimes by its full name, ubiquitone, but it's going to receive the electrons and pass them along. FADH2 comes right here. So while NADH comes to the first complex, FADH2 comes to the second. FADH2 is going to also give up its electrons. Those electrons are going to be picked up by that Q again, and all of those electrons get pushed to this next complex, complex 3, which then pumps the protons out here, again, into the inner membrane space. The electrons then continue their traveling this way. So here's this thing called cytochrome C, or site C here, which is, again, a shuttle. It's bringing the electrons to number 4. And notice that we have no NADH over here, but we do have electrons, and that the energy from these electrons are going to, again, pump hydrogen out here. The ultimate electron acceptor is oxygen. So down at the bottom here it says 1/2 O2 because oxygen is split. The molecule O2 gets split into 2 oxygen atoms, which then join with hydrogens that are present in this matrix area, forming water. So this is why oxygen's necessary for cellular respiration and will drive this reaction to the end, which you see here, meaning that now we've freed up these NAD pluses and FADs to go and do the same thing again. This is the last piece. So what we just looked at was the electron transport chain. Now we're seeing the very final piece of this whole cellular respiration puzzle, and this is the role of this enzyme called ATP synthase, which is also embedded in that inner mitochondrial membrane. Remember that there's all these hydrogen ions, these protons up here, and this synthase acts as a motor and to create lots of ATP. And it does so by this activity that is called chemiosmosis, meaning that all of these protons are going to actually travel back down their concentration gradient through this membrane, or this ATP synthase, and they're going to then, that energy from that flow of these protons now is going to create ATP. So this picture actually pulls it all together. You can see what's happening with NADH and FADH2. You see the electrons moving along. You see where those protons get pumped out. And now you're seeing this gradient, this high concentration of these protons. It's going to move through that ATP synthase and generate ATP. If oxygen's missing from the equation, then none of that's going to happen. Matter of fact, we have to go all the way back to glycolysis to see what the alternative route is if there's no oxygen available. And this is one of those 2 routes. So we see lactic acid fermentation would happen if there's no oxygen, because we need to regenerate this NAD plus in order to keep the process moving. And so to do that, we'll take a pyruvate, and the pyruvate will get converted again into something else called lactate. That frees up NAD plus, which can return to this glucose here, go through the process of glycolysis, extracting or oxidizing those different molecules until we have oxygen present that can then take these NADHs all the way into the mitochondria to do the oxidative phosphorylation. And that is the